Dear listeners, welcome all of you who are interested in electroencephalography, EEG, and event-related potentials, ERP, techniques, and all of you who are interested in collecting, annotating, standardizing, storing, processing, sharing, and publishing data from electrical activity of the human brain. Welcome to the fifth part of our video tutorial that provides you with EEG data storage, sharing, and annotation. We will explain what metadata are and what they are good for. We will also discuss approaches, initiatives, and conceptual and technical solutions related to storing and sharing of EEG and ERP data and opportunities to publish them. For those of you who are beginners, we recommend first listening to the previous parts of our tutorial. The long-term value of collected data does not depend only on their current scientific significance, but also on the way the data are annotated, stored, shared, and eventually published. When we look again at our EEG data lifecycle, the green ellipses denote our current position in the lifecycle. We will talk now about their description, storage, sharing, and publication. Let's note that it is considered that not only analyzed data, but also raw EEG data can be suitable for publication. The advantage is, in this case, that these data can be later and independently analyzed by other scientists. Moreover, publishing raw data naturally leads us to think about their annotation, storage, and sharing more than when we know that only our team will use and analyze them in the future. When we already introduced our position in the EEG data lifecycle, we can now present an outline of the fifth part of our tutorial. First, we will describe importance and ways of annotating EEG data. Then, we will focus on data standardization efforts and initiatives in electrophysiology, and will continue with existing data formats for storing EEG and ERP data. But we will, of course, also introduce formats that are more general and allow us to describe and store electrophysiology, neurophysiology, or even general time series data. The next topic we will deal with is data sharing and publication. We will provide you with the examples of raw EEG data that were published. Then, an example of EEG data annotation within the Brain-Computer Interface Project Basil will be given. And we conclude this part of our tutorial with a short quiz. Description of experimental design and collected data, subsequent data storing, analysis, interpretation, and publication. These tasks are often provided by various people working in the lab. To provide a complete, clear, and not ambiguous description of collected data, in other words, to provide data annotation, is not an easy task for an individual scientist, and it is definitely a hard task for a laboratory with a constant flow of researchers incoming and leaving. Such annotation has to be clearly understandable not only to the experimenter who has collected data, but also to other scientists, analysts, or data users, and has to be clearly understandable not only now when the experimenter knows and remembers the circumstances related to data collecting, but will be understandable also in the future. Data and their annotation are thus very important for correct data representation and, of course, their current and future understanding and interpretation. When the data are only partly annotated, or even if they are not annotated at all, we can hardly interpret them. Then, for even basic data annotation, it is necessary to get information, for example from a recording process, surrounding environment, or tested subject. A simple example in the EEG context is the sampling rate with which the data are recorded. We can argue that this is not the data we should store. We naturally know it since we normally collect data at some sampling rate. However, what if data are considered to be shared in collaboration? At least at this point, this kind of information is essential to make sense out of the shared data. Of course, a complete description of the data includes much more, for example, stimuli parameters, measuring units, and so on. To describe the whole experiment and data collection process, it is necessary to store information such as the design of the experiment, identification of a tested subject, more detailed information about the subject, and so on. This kind of information guarantees that the stored data could be correctly interpreted and reusable in the future. Such improvement of data quality also reduces research costs. For example, it is not necessary to repeat the same experiment to get the data again. And which is also obvious, raw data can be repeatedly analyzed. All important conditions or circumstances under which a recording was done, an experiment performed or analysis made, and all important pieces of information we know about experimental design, data collection process, tested subjects, and so on, which are worth identifying and storing for future use, are called metadata. It means that metadata are in fact data about data. However, the border between data and metadata is not always clearly defined. Metadata themselves can be divided into subsets from different points of view. Common categories of metadata are structural, descriptive, and administrative. Structural metadata describe the structure in which data are stored, for example, a table header. Descriptive metadata describe the nature and origin of data, and administrative metadata include, for example, a creation date of some file. In the case of EEG recordings, we can consider raw brain waveforms as data. The structure in which these data are stored, commonly binary data representation, is described by structural metadata. Descriptive metadata contain information about any step of data processing, typically data acquisition and analysis, and provides knowledge necessary to provide a correct data interpretation. Descriptive metadata are, for example, the number of electrodes, electrode impedance, event timestamps, and age and gender of a tested subject. Nevertheless, there are also metadata related to the whole experiment, for example, environmental conditions, which are important for the broader context interpretation of the recording. Another view of metadata should be also mentioned. If data are considered as a potential analysis input, then metadata could be considered as an analysis input filter and/or an analysis parameter. There is a general question how to recognize which metadata, and of course also data, are important. It means which data or metadata are worth collecting and storing. We have to admit that the correct answer to this question might not be clear in advance. Although a basic set of data and metadata collected can be derived from the experimented objectives, other interesting scientific questions requiring a larger or a different metadata set can appear later. However, we cannot collect metadata just for all cases. In the case of personal data, in our terms metadata, we are restricted by the law, GDPR, and we can collect and store only such personal metadata that are necessary to achieve predefined scientific goals. There are several approaches to data annotation depending on related conceptual models and technical solutions. The conceptual view includes, for example, our needs to separate metadata into some conceptual subcategories, or if we are just okay with a simple list of metadata items. Another example is if we want to use some standardized terminological terms, or we do not mind using our naming of concepts. Of course, using any standardized approach can substantially increase the value of annotated data, and we will discuss data and metadata standardization initiatives later. The technical view includes, for example, the used data formats, data containers, interfaces, and already implemented software libraries. Some data formats use a special file containing metadata. Some data formats include this information in a single container. Metadata could be also saved in a database, usually non-relational, or just written into a text file, or in the worst case on a piece of paper. Now it is time for an example of EEG data annotation. We use a brain vision format to store data and a small set of metadata from the experiments conducted in the neuroinformatics laboratory at the University of West Bohemia. Although this format is proprietary, its specification is freely available and can be processed using common tools, for example MATLAB or Python tools. It consists of 3 files. An EEG file contains raw EEG data in a binary format. Metadata are included in the brain vision header file, VHDR file, and brain vision marker file, VMRK file, while the VHDR file contains basic metadata related to the measurement setup, such as the sampling rate, used channels, their initial impedances, amplifier setup, and used software filters. The VMRK file contains detailed information about markers, stimuli, and responses to them, such as marker identification, type, description, its position and size in data points, and its relation to channels. An example of the structure of an VMRK file is also clearly visible from the slide. Some metadata from the VHDR file are also automatically extracted and transferred to standardized metadata structures. There are plenty of converters from the brain vision format to other formats to store electrophysiological data. The members of the Neuroinformatics Laboratory at the University of West Bohemia have used this format to store experimental EEG and event-related potential data in the EEG ERP portal, EEG base. This format has been also used for data publications in international journals. The brain vision format allows us to store a limited and predefined set of metadata. Moreover, these data are commonly stored in folders on the disk. It means that we need another tool that facilitates further organization and annotation of EEG data. The EEG ERP portal, also called EEG Base, is a web-based application for storage, annotation, management and sharing of experimental EEG ERP data and metadata. It gathers all EEG or event-related potential data and information about them collected in the Neuroinformatics Laboratory at the University of West Bohemia. EEG data are stored as binary files in the brain vision format, together with VMRK and VHDR files and portal administration data. They are stored in the relational database. Metadata, due to its flexibility, are stored in a non-relational database and can be organized using an ODML structure and terminology. The ODML structure and terminology will be discussed later. This approach allows us not only to annotate and organize experimental data, but also to search quickly for a specified measurement or filter measurements using predefined criteria. Possible disadvantage is that EEG base is not a complex tool and EEG data cannot be further processed in the portal, since the portal enables users only a very basic set of analytic tools. Thus, it is necessary to download data to a local computer to process them using another analytic tool. To annotate EEG data practically, EEG Base allows experimenters to create custom metadata templates that have a form of tree structures and use the ODML terminology. This terminology can be easily and freely extended to save all obtained metadata related to the measurement, experiment, tested subjects, conditions, analysis, and so on. The metadata are flexibly organized in subcategories, also sections according to the ODML model. Moreover, in EEG Base, newly created terms and templates do not have to follow or finally extend ODML terminology. You can just create your custom terms and structures. The already created templates can be easily modified and renamed, so creating a new template based on an existing template is even easier. The metadata structures can be extracted from EEG Base as XML files. The way we annotate data in our laboratory indicates some difficulties that are related to description and standardization of EEG and event-related potential data. On one hand, we need some standardized data formats, metadata structures, and terminologies to successfully and meaningfully annotate and share data in a longer term. On the other hand, experimental work is creative and we have to cope with the situation that new terms are needed to be defined and new metadata structures are needed to be created. Moreover, appropriate data annotation is a time-consuming process and experimenters will hardly annotate their data properly, not seeing the future benefits and not having appropriate and flexible tools. In electrophysiology, there is currently no standardized way to collect, store, annotate, and share data among researchers. The lack of standardized formats and terminologies has made sharing, exchange, comparison, and replication of data across laboratories difficult and almost impossible, significantly slowing progress in the field. Fortunately, the situation has started to change, and the need for standardized but still flexible data and metadata structures and terminologies supported by easy-to-use software tools resulted in the conceptual definition of standardized data descriptions and the development of related data and metadata formats and tools. The following slides are thus dedicated to standardization initiatives in electrophysiology and to widely used as well as new and promising data and metadata formats and terminologies. International Neuroinformatics Coordinating Facility, INCF, supports and develops collaborative neuroinformatics infrastructure and promotes the sharing of data and computing resources to the international research community. The INCF program on standards for data sharing was formed by the INCF with the key aim to develop generic standards and tools to facilitate the recording, sharing, and reporting of neuroscience metadata, in order to improve practices for the archiving and sharing of neuroscience data. It was operated in first two phases, from 2006 to 2010 and from 2011 to 2015 of INCF. The key deliveries of the program within electrophysiology task force were, for example, requirements for storing neurophysiology data, proposed HDF5 standard for storing electrophysiology data, further elaborated within the Neurodata Without Borders initiatives, and Ontology for Experimental Neurophysiology, OEN. Neurodata Without Borders is an initiative, consortium of researchers and supporters with a shared interest in breaking down the obstacles to data sharing, and also the project that has the aim to develop a unified data format for cellular-based neurophysiology data. The supporters involve, for example, INCF, Kavli Foundation, General Electric, Allen Institute for Brain Science, the Howard Hughes Medical Institute, Genelia Farm, the Simons Foundation, and the University of California, Berkeley. The overall aim of the consortium is to standardize neuroscience data on an international scale. It means to break down the barriers that restrain the cooperation and data sharing within the scientific community. Neurodata Without Borders has been undertaking a series of pilot projects. The most important one for us is the project related to neurophysiology that aims to develop a unified, extensible, open-source data format for cellular-based neurophysiology data. Now, we will focus on existing data formats for storing EEG and event-related potential data, but we, of course, also introduce formats that are more general and allow us to describe and store electrophysiology, neurophysiology, or even general time series data. The first introduced format is not actually a specific format. It involves a group of formats that just simply use binary and text files to store EEG data. While metadata could be stored as text files, data could be stored both as simple text files or binary files with a defined structure. These formats are, due to their simplicity, widely used. Some of them are proprietary and some of them even closed. A typical example is the already presented Brain Vision format that strictly separates data file from metadata files. On the other hand, another file-based and widespread format, EDF and EDF+, combines data and metadata in a single file. On the following slides, we present a representative selection of these formats. A well-known and widespread format for storing EEG data and metadata together in a single file is the European Data Format, EDF, and its extension EDF+. EDF was already introduced in 1992 and, due to its simplicity and openness, started to be widely used for EEG data storage and exchange. In general, EDF is a format for storing multi-channel biological and physical signals. The original EDF format had several limitations. It could handle only uninterrupted recordings and was not suitable for storing event-related potential or cardiology data. EDF Plus was specified to standardize most of the labels and add a possibility to save annotations and analytic results. The EDF Plus format thus supports annotations for texts, events, and stimuli. Currently, the EDF Plus specification is incompatible with EDF only in allowing storage of several non-continuous recordings. EDF or EDF Plus data file consists of a header record followed by data records. The variable length header record identifies the patient and specifies the technical characteristics of the recorded signals. The structure of EDF information about signals, subject, and whole measurements is defined and could not be changed. The EDF Plus format includes, for example, a version of the format, patient identification, record identification, number of data records, duration of records, and number of signals in the data records. All information in one single file is on one hand an advantage of the format. It is a simple solution. On the other hand, all information in a single file violates a basic rule of data management: separation of data of a different nature, data and metadata in our case. The disadvantages are then, for example, definition of terminology within a fixed structure, inability to define metadata subgroups, or possible performance difficulties when reading the data records. However, we have to admit that this approach, putting all information together, has appeared again in the form of so-called data containers. However, the inner structure of containers and their use via interfaces is optimized. Extensible Data Format, XDF, is an open-source community project and general-purpose container format for multi-channel time series data with extensive associated metadata. Except biosignal data such as EEG, EMG, EOG, it can handle data with high sampling rate or data with a high number of channels. Metadata are stored as XML. The XDF concept and beta version was developed by Christian Koch and Clemens Brunner at the Schwartz Center for Computational Neuroscience, SCCN, at the University of California, San Diego, USA. An XDF file can contain any number of multi-channel streams, each of which comprises an XML-based header that specifies both format and metadata of the stream, as well as the data samples. The organization of data and metadata types is similar to the EDF and EDF+ format. This format was already described in this chapter. It is a proprietary format with open structure that is primarily used in the tools of the Brain Products company. In this part, we only emphasize that also this format uses binary files for raw EEG data and text files for metadata. Comparing to EDF and EDF+, we can see the data that are stored in an EEG file are separated from metadata that are stored in VHDR and VMRK files. Moreover, definitions or events and stimuli in VMRK file that are necessary for event-related potential experiments are separated from metadata describing the recording process, VHDR file. In summary, this format allows better separation of data and metadata parts, including metadata subgrouping in text files, but the organization of metadata is still not flexible. Now we move a little bit from file-based formats to more complex solutions. NEO is a software package in Python for representing electrophysiology data. It is developed by the Neural Ensemble community-based initiative. NEO can load and convert data from closed manufacturers formats as Exxon, Spike2, Alpha Omega, BlackRock, and others. It provides and implements a hierarchical object data model consisting of 14 classes and finally supports exporting of proprietary formats to several widely used formats, such as HDF5 or MATLAB matfile. These formats then include a specific NEO structure, for example, NEO HDF5 or NEO MATLAB. As a result, interoperability between tools for generating, analyzing, and visualizing electrophysiology data is improved. The recommended way of using this package is to convert a closed format into an open format. The classes of the NEO object model are divided into three types: data objects, container objects, and grouping objects. While data objects contain numerical data together with associated metadata, container objects provide a simple hierarchical grouping of data, and grouping objects express the relationships between data objects. It is important that any object may contain any number of additional attributes or annotations that take the form of key-value pairs. Such annotations may be of various types: numerical or text values, arrays of such values, or dictionaries, hash tables containing such values. The idea of the NEO object model is related to the idea of the NIX and ODML models that are presented on the next slides, but NEO is more domain-specific. The NEO object model represents the basic data objects related to the electrophysiological recordings and provides the other two types of objects that represent a typical structure related to electrophysiological recordings. Metadata are associated to any class and can be thus flexibly added and maintained, but they are not organized in their own hierarchical structure. The NICS project started in the context of the INCF Electrophysiology Task Force, and NICS models for data and metadata and their subsequent implementations were provided by the German neuroinformatics node. NICS also supports the idea of separating data and metadata, but it goes much further. Since its data and metadata models are minimalist and highly generic, they are able to represent data stored in other widely used formats or models, for example in NEO, without any loss of information. It also means that both data and metadata model are not specific to the electrophysiology domain, but can be linked to predefined or custom terminologies to give elements of the models a domain-specific context. NICS also uses the ODML terminology for metadata description and the HDF5 file format as a storage backend. Last but not least, NICS provides libraries to simplify the access to the defined format. The NICS model for data consists of 6 main elements: block, data array, tag, multi-tag, source, and group. Multi-tag is not explicitly depicted here. A simplified NICS model that you can see on the slide demonstrates the basic entity types that describe a dataset containing an annotated analog signal. In summary, NICS provides separate models for data and metadata, but finally the data together with its metadata are stored within the same HDF5 container. The NICS data model provides a generic structure for description of annotated data arrays that can be easily linked with terminologies, providing together a suitable data structure for the electrophysiology domain. The NICS data model is linked with the metadata model that was also mentioned on the previous slide. The model for storing metadata is largely equivalent to the ODML, Open Metadata Markup Language, model that is also an initiative of the German neuroinformatics node. Now, we focus on ODML a little bit more. ODML is a file-based format and possible approach for annotation of electrophysiology data when we need a predefined and preferably modifiable and extensible terminology, and a data model for flexible organization of terms. The ODML data model that you can see on the slide is based on the idea of key-value pairs and is highly generic as in the case of the NICS data model, and thus defines and establishes an open, flexible and easy-to-use format to organize and transport metadata. The metadata model itself defines 4 entities: property, section, value, and root section. Properties containing values can be grouped into sections, which themselves can be nested to build a tree structure. The root section of the tree contains some administrative metadata. It means that ODML itself defines the model, but not the items that are described or the terms that are used. To be domain-oriented, each section can be based on an ODML terminology that provides a standard, but does not restrict a possible definition of other terms within the terminology. Finally, data and metadata have to be linked and integrated. Within NICS, the major entities of the data model, it means block, tag, data array, and source, contain the metadata field, which is used to create a link between data and metadata. The Neurodata Without Borders data format is based on representative use case studies from 4 laboratories, and the project includes a vetting phase for assessing whether other data models can also be used in the new common format. The resulting NWB format is designed to be flexible to incorporate several kinds of data, including electrophysiological and optical physiology data, and complex metadata related to stimuli and behavior. The Neurodata Without Borders format uses the hierarchical data format, HDF5, as primary storage mechanism. The storage scheme is described by a specification language, and it is extensible to support future scientific needs as well as lab-specific requirements. Data are organized hierarchically using easy-to-use primitives, for example groups, which are similar to folders, datasets, and dimensional arrays, attributes, which are metadata objects on groups and datasets, and links. These are links to groups and datasets. Beta version 2.0 of the format as a format specification, application programming interface, and a list of tools to work with the NWB format is also available. Neurophysiology sample datasets contributed by 4 laboratories have been translated into the NWB Unified Data format. Comparing to other formats, the Neurodata Without Border format provides a very detailed format specification, including the used terminology. The overall idea is similar to the NIX and ODML formats. With the previous slide, we have finished the basic overview of existing data and metadata initiatives, descriptions, approaches, and formats. Now we will continue with the next block of our lifecycle diagram, data publishing. At the beginning, we have to emphasize that we consider publishing raw or analyzed data as an essential step during publication of research. This approach to publishing scientific results has not been very widespread, but this view, together with supporting standards and technical means, is gradually coming to the attention of authors and publishers. In our case, we will focus on the first step, that can be even more unpopular or even considered as dangerous among scientists: publishing of raw data. Scientists are often afraid that their colleagues can find scientifically interesting context in their data and publish it sooner than the team that collected the data does it. On the other hand, publishing raw data may not provide only raw material to scientists who are able to interpret them. It can also give a credit to the scientific teams providing such data, of course, if they are properly cited. Thus, let us consider a dialogue about data with other researchers as a beneficial step. Now we describe and generalize our experience with publishing raw EEG or health-related data in international journals, more specifically in Gigascience, Data in Brief, and Scientific Data. Our research objectives were various, for example, to make any conclusion about developmental coordination disorder in children from their EEG data, or to collect a large set of heterogeneous health-related data which could be used as a forewarning about various chronic diseases. Research objectives are important to be declared in the data article, but we also came to the situation that the greatest interest in the data was finally just a side effect. To describe and validate data, we used only basic statistical methods, mainly linear regression model. The publishing process itself consists of several steps. Many of them are similar to common publication process. The first step is searching for a suitable journal that is willing to publish EEG or event-related potential data. Since these data are still relatively rare and not easy to collect in large quantities, the journals we have already mentioned have interest in them in general. In the next step, a predefined publication template has to be followed. These templates are relatively detailed to provide authors with enough help to describe their data sufficiently and provide other necessary information. For example, to show an approval of ethics committee for the acquisition and publishing of the collected data. The next step differs from a common publication process. Not only the paper, but also the data and related metadata have to be stored and provided to reviewers. In this case, we used our EEG base for data storage and annotation, and mirrored the data in the database the journal required or recommended. The next step is the review itself, usually running in several iterations. Reviewers express their opinion not only on the quality of the concurrent descriptive paper, but also on the quality, attractiveness, and completeness of provided data and metadata. When the data article together with the related data and metadata is accepted for publication, the final data curation service is provided by the publisher. There are a few other things that are good to know. There is a difference when you publish a scientific dataset that contains data only from one measurement or a clinical trial registration record that contains each participant data from repeated measurements. In the latter case, you have to pay more attention to security, description and registration of collected records. Informed consent and approval of ethics committee are required. Data licensing policy is defined by each journal, but various types of Creative Commons license are required or at least recommended. Our research group has published collected data and metadata in 3 journals: Gigascience, Scientific Data, and Data in Brief. while the publications in gigascience and scientific data journals deal with event-related potential data, the publication in Data in Brief deals with other health-related data. To search and manage your publications, the services such as Google Scholar, ResearchGate, Researcher ID, and ORCID are beneficial to use. The Brain-Computer Interface Project Basil was already introduced in the previous parts of our tutorial. Here, we only would like to remind you that the goal of this brain-driven computer assistant system is to provide communication and control pathways to people with severe motor disabilities. Currently, the EEG and ERP data collected within this project are stored in our EEG base in their original, it means brain vision format. However, the data are annotated according to the ODML structure and terminology. Specific examples of annotated datasets you can find in the supplementary material to this tutorial. Now we are at the end of our 5th chapter and we have again time for a quiz. In the following questions, only one answer is incorrect. Why is important to annotate data? A. To improve information value of data. B. To follow lab requirements. C. To preserve important information for future use. The incorrect answer is B. What does the term metadata mean? A. Additional information describing the nature and origin of data. B. Data that are expressed at a higher abstract level. C. Description of the circumstances of the experiment. The incorrect answer is B. Which technical solution is usually not used for storing an EEG record? A. HDF5 container. B. Single file. C. Relational database. The incorrect answer is C. What is not part of EEG metadata? A. Data related to the tested subject. B. Raw EEG data. C. Information about recording channels. The incorrect answer is B. To publish an EEG ERP data article, it is necessary to A. Use the EDF plus format for the data. B. Specify the type of license. C. Annotate the data. The incorrect answer is A. Our quiz is over, and now we can take a short look on references. And we would like to invite you to the last, 6th part of our tutorial that is dedicated to EEG signal processing and its applications.